Now let us see about topic DNA barcoding. What is DNA barcoding? The DNA barcoding is a taxonomic method that uses a very short genetic sequences from a standard part of the genome is called DNA barcoding. Have you seen how scanners are used in supermarket to distinguish the universal product code? The universal product code UPC. In the same way, we can also distinguish one species from another. Then, the DNA barcoding is a taxonomic method that uses a very short genetic sequences from a standard part of genome. The genetic sequences used to identify a plant is known as DNA tag or DNA barcodes. The genetic sequences used to identify a plant is known as DNA tag or DNA barcodes. Then the DNA barcode, the proposed by Paul Gabart in 2003. So, the Paul Gabart considered as father of barcoding. The Paul Gabart in 2003 proposed the DNA barcoding. So, the Paul Gabart is considered as father of barcoding. Then, the two gene region that is being used as an effective barcode in plants is present in two genes of the chloroplast. The two genes present in the chloroplast. First one is MATK and RBCL. The MATK means megakaryocyte associated tyrosine kinase and RBCL. RBCL means the ribulose bisphosphate carboxylase, the large chain. The two genes the present in the chloroplast and have been approved as the barcode regions for plants. The two genes also have been approved as the barcode regions for plants. Then, the sequence of unknown species can be matched from submitted sequences in gene bank using BLAST. BLAST, basic local alignment tool. The sequence of unknown species can be matched from submitted sequence in gene bank using BLAST. The BLAST web program for searching the closely related sequence. Then, significance of DNA barcoding. The first one, DNA barcoding greatly helps in identification and classification of organisms. Then second one is, it aids in mapping the extent of biodiversity. Then next one is, DNA barcoding technique requires a large database of sequence for comparison and the prior knowledge of the barcoding region. However, the DNA barcoding is a helpful tool to determine the authenticity of botanical material in whole, cut or powder form. Then differences between classical taxonomy and modern taxonomy. Now see the first one is classical taxonomy. The classical taxonomy, it referred as old systematics or alpha taxonomy or taxonomy. The modern taxonomy, they referred as neo-systematics or biosystematics or omega taxonomy. The modern taxonomy, the alpha taxonomy, they changed to omega taxonomy is called as modern taxonomy. The classical taxonomy, it is pre-Darwinian. The modern taxonomy, it is post-Darwinian. The third one difference is classical taxonomy. The species is considered as basic unit and is static. The modern taxonomy, the species is considered as dynamic entity and ever changing. Then next one is classical taxonomy. The classification is mainly based on morphological characters only. Then earlier the classical taxonomy, the classification is mainly based on morphology characters. The modern taxonomy, the classification based on morphological characters, reproductive characters and phylogenetic characters. Phylogenetic means evolutionary characters, the relationship of the organisms. Then the classical taxonomy, this system is based on the observation of a few samples individuals. The modern taxonomy, this system is based on the observation of large number of samples and individuals.